Um, right. If you look at the title giving is towards the solution of protein structure prediction problem, which I think is appropriate title because we are now really closer than ever in the history to solve the protein structure prediction problem, which mainly is due to the uh, introduction of the AI and deep learning method. So before I touch about the core technology, I will spend some time to introduce the background of the field and also the previous effort of the of this field on the classical method and approach. So that will make us better, appreciate the really excitement and the progress of the field. So this is a slides which I taken from my teaching class. It's called sequence to structure to function paradigm. So basically, you start from a sequence, then you try to decide the three-dimensional structure, and then you can annotate the function, and one of the final goals is for drug discovery. So here, from the sequence to structure, that is a major, the topic which of the structure biology for protein structure determination. And when we talk about the protein structure determination, there is really a long history for the structure biology. The first milestone probably can be traced back to more than 100 years ago, where Dr. Zhang discovered X-ray, and he deserved the first Nobel Prize in the physics because of discovery. So in 1921, and then Dr. Wong Loi, he found the X-ray. When the X-ray cross the crystal, then you can have the diffraction pattern. So from the diffraction pattern, we can trace back to determine the location of every atom in the crystal. So that build up the structure, the method or background for the protein structure determination. So based on that method, and Dr. Zhang solved the first ever the protein structure is a myoglobin in the 1958. So that moment, every of the protein structure is solved, it's a big deal. So probably you will grant the Nobel Prize because it's so important because the human being has first time to view how the protein small molecule looks like. So in 1971, they built the protein data bank. So that moment we only have 2 protein structures. So come to 1980, then we have 184 structures in the protein data bank. So 1985, so NMR come to the game. So about 10 years ago, there's a very important method, it's cryo-EM. They are now, solving a lot of the protein structure, which is, especially for the membrane protein and the protein complex. So if I look at by yesterday, and we found in the protein data bank, we have nearly 200,000 protein structures. So if you look at the number of the structure, they are growing very fast. So every day we have about 35 new protein structures solved. So it's very encouraging. But if you compare that protein structure with the protein sequences which we know in the data bank, we will be very frustrated because the gap in 2007 is only 100 times larger of the sequence compared with the structure. So until now, it's more than 1,000 larger. the gap is become larger and larger. So that is very frustrating because the number of structures can never catch up the sequence. But we know that sequence cannot tell us too much about the function or help us for the drug screening. And structure is so important. So what we can do? So fortunately we have a computer and we can, so the goal of the protein structure prediction is to try to develop a computational method to predict the three-dimensional structure from the amino acid sequence. So this is, as we know, the major challenge in the modern computational biology. So when I prepared those slides quite a long time ago, we are questioning that whether it's possible. Now it's for sure it's possible to predict the structure. So it's just as Sebastian just mentioned, we are developing methods called iTaser, and at that moment we are mainly based on homologous modeling and also assisted by the physical-based Monte Carlo simulation. So we start from a closed sequence. We try to identify the structure template from the PDB library by threading. Then after we get the template, we break the template into fragments based on the alignment. So here you can see the thick backbone are aligned region, the thin backbone are unaligned region. Then we align those fragments because the local fragments have pretty high accuracy. So then we use Monte Carlo simulation to assemble the fragment into full-length model. Then we use clustering to identify the lowest free energy based on the largest the cluster is the lowest free energy because this corresponding to the partition, the biggest partition function. So then we get the final model, or not the final model, it's a cluster centroid. And then in order to refine the detail of the local atomic fragments, then we reassemble them using a more like a atomic level force field. Then we get the final model. If we have the final model, then we can, annotate the function by compare the structure and the local and global structure with the function library. So that is the method. And the first field is mainly a knowledge-based first field. We have major first sources. One is the statistical term derived from the PDB library and include hydrogen bonding, and also the propensity to the predict secondary structure. Also the hydrogen bonding is a very important part, and hypotheticity using neural network training. So very important term is the homology based from the template derived the potential. So there's overall about 26 terms. One of the very important questions is that how we can combine them. A very simple way is combine them by linear combination. But those energy terms are not independent. So we have to find an optimized way to optimize the parameter of the weighting. So to do that, we are using a decoy-based optimization. So we generate many decoys, and then we try to optimize the correlation between the arm speed to the native and the first field, total energy. So if we naively set all the parameters as 1, you can see this first field is not good because the lowest energy is quite far away from the native structure. But after we optimize the weaken factor, we can see there's a better correlation. And so when we test our method, that moment, we have a very standard method. It's called modeling. It's widely used compared to modeling. So we are starting from the same set of the template. We found that we can fold about 2/3 of the proteins. and the model can also is good. It's a 44%. So the question is that why there's another 1/3 we cannot afford at the moment. So to answer that question, we did a very simple, experiment. So this is a pipeline of the iteration. Then we don't start from the template. We just start from the best possible template. Suppose we know the protein structure, native structure. Then we use the structure comparison, search the PDB library to find the best template. Then we start from here, try to assemble the structure. Very importantly, we remove all the homologs which have a sequence identity higher than 24 or 25%. So the result is quite surprising because we found that if we use the best possible template, then we are able to fold almost all the protein structures. So the average RMCD is 2.25 angstrom. So that is one of the earliest date which we concluded that the PDB library is complete for us to solve the protein structure prediction problem. So this data is still useful for nowadays, even we have the deep learning or AI technology, because this tells us the template is already there. So if we can use a deep neural network, we can probably use a network to find the better template. Then we can also use another strategy or avenue to solve the problem. But the major issue is that we are not able to identify the better template, because if you are starting from a sequence, you do, real life threading, we are only be able to fold this, this number of the protein we can fold. So that is the template-based modeling. And another method we developed is called Quark. That is, it's developed by a very smart student of my lab. And he started from a query sequence and then identified some very short fragment from the, unrelated protein structure. And then using that fragment, try to reassemble the structure. So this fragment is different from ITASA because this fragment is not from the homologous templates. But he invented a very interesting technology, is that he is able to identify the contact map from the fragment. So his idea is like this. So suppose you have a query sequence. Then let's look at the two residue I and J, two positions. And then for residue I, you can identify a number of the fragments. And residue J, you also identify a number of fragments. And if this some fragment and this here and here, they are coming from the same protein structure, then we are able to calculate the distance from this for Dij. Of course, we can calculate a number of these distances. Then we can, plot it's called a profile, using the profile. So most of the cases is trivial. You can see they are like diverse. But there are some cases you have a peak in the, like a Gaussian in some limited distance. So that probably tells this distance is true, or these two residues are in contact. So in that way, we are able to use Quark to fold many of the protein which have very complicated topology. So those are some of the examples. And the TM score, compared with the Rosetta, they are doing, in most of the cases, or on average, also they are better. So those are the two methods which we have developed based on template and the ab initio folding. So certainly we are not alone. There's many of the other laboratories in the world, they are developed different methods. So this is the name of this, the PI and the institution, the software name, and those are different methods they are using. So there's one question is that what method, really work, what method doesn't work, because those different methods, they're published separately in different literature. So to answer that question, just Sebastian just mentioned, there is a Olympic game in the field. It's organized, it's called a CASP. So every 2 years, the organizer collect about 100 protein sequences, which nobody knows the structure. Then they send the sequence to the predictor, asking the predictor protein structure. In the same time, they will ask these structural biologists to solve the structure using X-ray or NMR or other cryo-EM. And then 3 months later, there is a third party referee, the scientists. They will compare their predicted model with the experimental structure. So, and then they will have a very objective way to assess the accuracy of the model. So you can see this is one of the examples. The native structure is red and the predicted model is blue. So you can calculate the exact error in every location of your prediction. So this is probably the first, earliest, the competition which is introduced to scientific area. So that is the first test which started from 1994, and every 2 years. So there's a human prediction group. There's also solar prediction group. Solar prediction means your prediction is fully automated. You build up a solver and then organize a submit a sequence to your solver, then automatically generate the prediction. So human, you have a longer time. You have 3 weeks, and the solver you have to generate your prediction like in 72 hours. So our lab participated in the 2006 CASP7, and we are actually participating every time. So I will mainly focus on this last 5 CASPs. I will overview that because those are very typical, because the CASP 11 probably is the last classical approach, and the CASP 12, there's new technologies called co-evolution. The last 3 CASPs is really dominated by the deep learning. We will see how deep learning really changes the field. So in CASP 11, these are the numbers. So there's that many of the human expert groups, this is solver groups, and the number of the protein targets. The target is split into 2 categories. One is the template-based modeling. You have some template in the PDB library. So you also have the more difficult cases. You don't have any template. So for the template-based modeling, your goal is first you want to identify the best template. Second, you want to refine your model, your template closer to the native. So this is a native structure, and this is your template, your final model. You have to compare which one is closer. So that is actually a very challenging problem, and for many years, and the template either make the template worse, or you are just keep the template unchanged. So this part is one of the first time we can see the final model. This is MT of the first model, and this MT of the template. So you can see many of the points. One point is one protein target. So many of the points, they are below the diagonal line, means our, the modeling draw the template closer to the native structure. So this is a TM score. TM score is higher the better. So majority of the templates are better in the, the model is better than the template. So what about the free modeling target, which we don't have a template? So we have to build a first field and use a search engine to find the lowest free image based on the Antonsen's assumption. So
hopefully the final, the lowest free energy, we can identify the template, the better model. So here the goal is that because our phosphate is not an athlete, the goal is to construct a correct fold from scratch. If your TM score higher than 0.5, that means your model is have the correct fold. So these are probably two of the best model which we have built. One is this is T0604. This is in the class 9. This is the class 11. So this is a X-ray structure and the predictor models are very close, and we don't use any template field. So let's look at this the Cas11 target, and this is a TM score. This is the length of the protein. So before Cas11, there's no target we can fold which have a size larger than 100 amino acid. So this is a target which we are very proud of because we build a model which is much higher than 0.5. And so if we look at the template-based modeling, the iTaser, we start from this fragment and then build the model. This N-terminal is a mirror image, it's wrong. But when we use quark, which uses the fragments, and then we correctly predict the middle range contact map in this N-terminal. So we are building a better model and compare with the native structure. So the RMC is 2.4, TM score is 0.736. So what about this? That is highlighted about Cas11. So in Cas12, the very important, if you look at this comparison, we can see we have much larger number of this target which generate the correct fold, have a TM score higher than 0.5. And if you look at the folding rate, here is 10%, now we increase to 34%. Why we make it so big, the progress? The major reason is that we have using a method is called co-evolution to predict the contact map that guide us to generate a better model. The idea is very simple. So from the query sequence, we construct a larger number of these homologous sequence. We have a multiple sequence alignment. So the co-evolution means if you have a residue here, when they mutate, this residue in this beat, they also correlately mutated. For example, you have L is not mutated, then W is not mutated. If L mutated to W, W mutated to E. W mutated to K, so this E will mutate to V. So there's correlation in this mutation. That probably means they are in three-dimensional space. They are also in some way they are in contact. Because they are in contact, so when you have one mutation in one position, you have another position, you can make also correlated mutation so that you can compensate this, this spatial change, make the structure stable. So that idea is actually proposed quite a long time ago. It's probably the first appeared in 1994. But they are not really very helpful because there's a transient noise. Because some cases, for example, if A and B, they are correlated mutated. B and C correlated mutated. But then A and C will also correlate mutated. But you can see A and C are not really in contact. So that is kind of transient noise which reduce the accuracy of the contact based on the co-evolution. So we are using a technology called directly coupling analysis. So here, this is a conformational probability will be can be written in the form based on the parts model, right, in this way. So here, this coupling, pairwise coupling, can be associated directly with the contact map. That pairwise, coupling can be derived directly from the pairwise frequency parameters, co-evolution parameter. So that will significantly increase the accuracy of the contact map. That's a major reason for this improvement. So here we are introduce the contact into the quark, and then the result is still that we have 8 proteins target. We have correct fold. This is a native structure. This is our predictor model. The success is mainly because of contact. 5 cases, the contact map accuracy have a very high accuracy, so we can know these are because of co-evolution. So that is highlighted in case of 12. What about the case of 13? So in case of 13, and again we look at the progress. Now we can fold, 72% of the cases have a GM score higher than 0.5. So the major weapon is because of deep learning. We are still derive the co-evolution, derive the contact map, but we are not using the DCA. We are using deep learning to learn the contact. So this is a pipeline which we developed by a very smart PhD student in my lab. He developed a method called REST-free. So start from the multiple sequence alignment, he used the residual convolutional neural network to directly predict the contact map. And different from others, he's using a precision matrix is a inverse of the co-evolution matrix. So his accuracy in the benchmark test is much higher than all other different methods. This SVM SEQ, this is also deep learning. It's not deep learning, it's a machine learning neural network, which we developed more than 10 years ago. So you can see they are dramatically changed, the deep learning. And those are in the case, there's also a section for this contact map. The triplet rest is a newer version for the REST-free. So because of this much improved contact map, we can fold a much larger number of this fraction of the target. So we can fold 32 free modeling target with a correct fold. So in case of 14, that is we are still using a deep learning, but we are using a more comprehensive pipeline to predict not only contact, also predict distance and hydrogen bonding network and orientation, and integrate these restraints into the ITaser. So when we predict, when we use deep learning, we are predicted is a histogram of the probability. So how we convert that to the ITaser, combined with the ITaser first field, we are using this, the negative logarithm of this probability. So we use some trick to combine different range of the model, which we found very helpful. So we are not going to that detail, but we can enjoy this very dramatic, surprising change. So this is a classical ITaser. This is a D-ITaser when we use a more comprehensive deep learning model. So you can see there really have almost all the targets we have improved, especially those star this free modeling target. We are able to convert those free modeling targets into the accuracy which is as good as the template-based modeling. So those targets is really don't use any template, but their accuracy is as close as the experimental, some low resolution experimental structure. So are those targets which we are successfully fold, and those are the targets which we can say majority of the cases we can fold the correct, fold. And those are just a compare of our solver. Down solver is iteration, quark is another method. Compared with other method in the case of 14, and actually, if we look at the top 10, so solvers, those are GDT score is the assessors use the score. GDT means if you look at this blue are the prediction model and the C alpha trace, red are experimental structure. So GDT score is look at the DI, any pair of the distance, check how many of the cases you have distance below 1 angstrom, 2 angstrom, 4 angstrom, 8 angstrom, and sum this number together, normalized by the length. So if your GDT score is good, then you get the GDT score, the perfect model is 1. So this score tells you how significant of your prediction model compared with the average prediction. So that is the case for 14. This is just the 8 cases, which from case for 7, 8, 9, 10 to this 14. So we're we're doing pretty it's reasonable, and especially when the new this is still classical method. Those are co-evolution. This these two cases we are using deep learning. So our gap is compared with others becomes smaller and smaller, especially when the new technology is introduced. So case 15 is I didn't participate. I participated in all this 8 cases, and because my lab is moving from the Michigan to Singapore. So, but one of my postdoc is whose name is Zhang. He participated using a new method called the DI tracer. So he's using the a very comprehensive way. It's called deep MSE to collect the multiple sequence alignment, and then use a multiple sequence alignment to train this distance map, and also use the AlphaFold, the end-to-end training template, integrate that into the iTaser, and then build the final model. So this result is even more surprising. So we can now fold 91% of the cases with a TM score higher than 0.5, and many cases our model higher than the TM score higher than 0.9. That means they're really close to the experimental structure. So those are some of the examples. So here is a comparison of his model with other prediction. So in the case of 15, there is 3 categories. One is the single domain. This is a traditional way. Now we also, case of assessor also look at the orientation and interaction of this domain-domain interaction. Those are multiple chains. So here is one chain. This is complex, contain 20 different chains. So surprisingly, their TM score is 0.94. So that means, although this is a very big complex, they don't have a template, but we are still able to build a model which is very close to experiments. So in all 3 categories, the Zhang's model is really ranked better than others, especially compared with AlphaFold. AlphaFold is very astonishing everybody in the case of 15, case of 14. But now there's a bunch of the methods doing better than test in AlphaFold. So that is really signifies that the really quick progress of the field, and especially in the domain, multiple domain or pre-screen interaction, the DI tracer, which can model better the orientation and the chain interaction. So here probably is one of the very surprising or the slides to, to highlight the progress of the field. In the previous, this is the best model we can build. The blue is the model, the red is the experimental structure. We can only fold the small alpha proteins using the classical method. When we use core evolution, we can build some bigger protein. But when we introduce deep learning, we can fold a much larger protein. And when we use more comprehensive deep learning, we can fold, first time we can build the free model target model. We can have accuracy close to experiments. This is in case of 15. We are model this huge protein, which is have 1,400 amino acid, and this is a single domain, very big. The chemistry is close to the experimental resolution. So we come to the conclusion, and the first conclusion is that the deep learning can fold nearly all the single domain proteins. That probably signifies that the problem for single domain protein structure prediction can be solved. There is a paradigm shift from the relying on the PDB based on homologous modeling. Till now, we only rely on the genome sequence, because from genome sequence, we can consider the multiple sequence alignment, then using deep learning to train this end-to-end training model. But we still have some challenge, because that strategy, deep learning, still relying on the multiple sequence alignment. If we don't have good multiple sequence alignment, for example, if you only have for some orphan protein, the deep learning still cannot work well. Second is that the metagenome, they are very helpful for the deep learning and MSA construction. But the metagenome data set increasing very fast. So nowadays, there's if we look at our comprehensive metagenome data set, the storage is more than 20 terabytes. So searching the entire database becomes more and more expensive. So how we can develop better program to collect the MSA from the metagenome in a quick way and accurate way. The last one is that we still have difficulty to model the multiple chain protein and the protein or protein RNA and DNA complex. So we have still, the strategy is that we are trying to combine the deep learning with a low resolution model with a cutting edge, like the structure assembly simulation. So here, I put a black box here in the deep learning, because sometimes we can predict the final model, but this network to us is the black box. We don't know what's going on inside of this. So that is really not satisfactory for the physicists, because the people really want to know how protein fold, what is the pathway in cell. So I think when we combine this, deep learning with the first field-based folding simulation, that probably can help us to understand not only the final structure, also understand the folding pathway. So before I end of my talk, I'd like to also introduce a very interesting work, which is performed by one of my PhD students. He, extended the end-to-end deep learning training, and also, they integrate this protein language model into the RNA structure prediction. So we found that this is the first time, we apply the deep learning into RNA structure prediction. So here, these are the RNA puzzle. It's a kind of test for the structure prediction. These are on the star of the Robbins model. You can see they are closer to the native than every of this prediction model in every of the puzzle. So those are the, the comparison of Robbins model with a native structure. Those are the best model from the entire community. Those entire community is mainly based on the traditional simulation method. So here you see one of the movie which can show that, using his method, the deep learning, he can fold a somehow I cannot show that movie, but anyhow, that one you can using a single laptop, similarly about in 1 minute, they can fold a middle-sized RNA structure from scratch into a well-folded shape. So anyway, this is the last slide I try to show you because those are which we are working on the problem. So this is what we talked today is protein structure prediction. We are also very excited about the protein design, which we try to design computational design new protein sequences beyond the natural protein. So we also work on the structure-based function notation and the protein-protein interaction. So one of the last one is we try to to model this single mutation and how that is related with the cancer and other human disease. I also want to thank this this funding support from the NIH and NSF. Now this is from the NUS, and thank you for the attention.